Hello, everyone. Welcome to the Gerontological Society of America and American Aging Association webinar, The Intersection of Aging and COVID-19. The webinar is being recorded and will be posted on the GSA website. A notice to all attendees will be distributed once the recording is available. Question and answer sessions will follow live presentations. So throughout the program, we will be accepting questions through the questions feature accessible on the GoToWebinar panel. Also located there are downloadable handouts for today, copies of presentation slides. So without further delay, I'll hand the microphone to Dr. Roz Anderson, President of AGE and GSA's 2021 Biological Sciences Vice Chair-elect. Roz?
Hi, everybody. I'm thrilled to be able to bring this webinar to you today. It's super exciting, not only because of the topic that we're covering, which is the intersection of aging and COVID-19, but what some of you may not know, that this is an epic event. 75 years of GSA and 50 years of the American Aging Association, and this is the first time we're coming together. So I'm really pleased to be able to reach across these two very long-lasting and go-to organizations for aging research. So our goal with the webinar is to really explore the basis for the skew in vulnerability to COVID-19. We know that older individuals are at much greater risk of poor outcomes once they succumb to the disease. And the session is designed to be translational. So we're spanning the spectrum of aging research in all of its different facets, which is something that I'm excited to bring to each of you who may be working in one discipline or another, but just to really showcase how multidisciplinary aging research is. We have 4 fantastic speakers. We're so lucky that they've given us their time today to come and provide their expertise and to offer opportunities for questions and answers. So you can, as Judy mentioned, you can fill in on the question box there and we can get through as many as we can. We'll have the 4 speakers give their presentations, a little bit of time for questions following each speaker, and then the remaining minutes of the session we'll do a panel discussion where it's basically a free-for-all. So without further ado, I'm going to introduce the first speaker, Tamara Konecka, who comes to us from the University of Chicago. And Tamara, I think I'll just hand it straight over to you in the interests of time.
Great, thank you. There we go. I'm very happy to be here today to talk about COVID-19 and older adults in long-term care facilities. I'm going to be focusing on the institutional and policy context and leave more of the biology to some of my colleagues. I should mention before I start that I'm also going to be discussing this mostly in the US context. I know we have an international audience on this webinar. Some of it will apply to other countries and some of it will not, and I'll try to note that as I go through. This is becoming an overused term, but the situation around COVID-19 in nursing homes and the number of cases and deaths that have accumulated has been described as a perfect storm of sorts. And I think of there being 3 main components of this perfect storm. First of all, there's the attributes of the novel coronavirus itself and how that intersects with the vulnerabilities of older adults. And then there's the institutional setting and how that exacerbates the problem. And then finally, there's the policy context. And I'll be talking a lot more about both the institutional and the policy context, but let me go through each of these in turn to make sure everybody knows what I mean by them. So in terms of the attributes of the coronavirus itself, of course, we now know that there's asymptomatic spread and that the virus is airborne. These were not well known by everyone at the outset of the pandemic. And everything we did in nursing homes prior to the pandemic, all the infection control procedures, which, even as they stood, were not always very well implemented, and there were a lot of known issues around infection control in nursing homes. But even the regulations we had prior to the pandemic were not really appropriately preparing facilities for this kind of a virus that could be spread asymptomatically through the air. So that's one problem. Next slide, please. And then there's the institutional context itself. So we know, obviously, almost by definition, that nursing homes house mostly older adults with underlying health conditions, and those are exactly the people who are most vulnerable to this virus. They also live in a congregate living situation. Often, the average size of a facility in the US is something around 100 beds, and often there are multiple residents per room. And so this is obviously the kind of situation that makes transmission of a virus very easy. The residents in those facilities need hours of hands-on care every day. So you can't really isolate them. Staff have to go from room to room providing care, and those same staff go in and out of the facility daily. And sometimes due to, mostly to low wages and the desire to earn a living wage, staff often work at multiple facilities. And then this is all exacerbated by the fact that most facilities in the US over the last couple of decades have suffered from chronic understaffing. This is due to a variety of problems that I'll touch on later, but I won't really go into in detail. But that chronic understaffing was exacerbated by the pandemic because the people who work in nursing homes, one may have gotten sick themselves, two may have, feared going to work or decided not to go to work because they didn't want, in the face of, shortages of personal protective equipment, didn't want to risk getting sick or risk bringing that virus home to their families. Or they had kids at home who were no longer attending school and had to be home with their kids. And so, all of what we know now is sort of best practices in terms of handling the virus requires staff to implement. And so this is another part of the institutional context that made fighting the virus and continues to make fighting the virus very difficult. Next slide, please. And then finally, there's the policy context. And in the US, what we have what I see as a dismal failure of federal policymakers to have secured the supply chains for testing and personal protective equipment. And this is a problem that's really still not solved. It's obviously the pandemic is a national problem, and the federal government has powers that no state or local governments have to try to force the production of those supplies that are desperately needed. And that didn't happen. This was sort of delegated to the states who had, limited ability to solve the problem. And what we see as the virus surges across the country yet again is that still, testing and PPE are inconsistently available and often in short supply just where they're needed most. And this is certainly true in nursing homes. This is true of the pandemic in general, but this is certainly true in nursing homes. That was compounded by the fact that long-term care facilities were really not prioritized early in the pandemic. There's been more focus on them in the past few months. But if we recall all the sort of media reports early in the pandemic, a lot of it was about hospital workers and shortages of PPE in hospitals, and that was a real concern and still is. But nursing homes were sort of at the bottom of that food chain, and so even as people worried about hospital workers, there were long-term care workers who were going into facilities and literally working with no PPE or just a homemade mask, despite the fact that they were working with COVID positive patients on a daily basis. This is in part because of the history of nursing homes in this country and the fact that they've often had a sort of not very positive reputation, and a lot of the industry is run by for-profit organizations. And I think there was a general reluctance. There's often a general reluctance by policymakers to assist what they see as a largely for-profit industry that should figure it out on their own, which of course in the face of a crisis may not actually be that helpful. So instead, what the federal government did is really focus on putting out guidelines, inspections, increasing inspections, and finding facilities that were not doing well fighting the virus. And of course, in the face of a crisis, these kind of more punitive measures really may not be helpful if the goal is to prevent cases and deaths. And it may actually hurt those facilities that need help the most. So there has been, some technical assistance and resources, funds that came out of the CARES Act, for example. But what's been sent out to the industry has mostly been slow, inconsistent, and inadequate. Next slide. So the result is, what we've all seen in the papers. There have been about 500,000 cases and more than 80,000 COVID-19 deaths in long-term care. There's also been obviously a number of staff cases, and there's a lower death rate, but still there's been almost 1,000 deaths among staff. One statistic that I find really striking is that approximately 80% of nursing homes have now had at least one case in this country. And so it's clearly not a bad apples problem, right? This is a crisis that was hitting all nursing homes, and it's not that some were, handling it so much better than others. It was a crisis for all of them and continues to be. Obviously, there's a high case fatality rate in long-term care, higher than in the general population, even though, the numbers are subject to debate. And so now we have about 40% of all COVID-19 deaths in this country are in long-term care, and many more in some states. Next slide. And so this just shows that there's variability across the states, and some states, looking at like Minnesota, it's 71%, right? So it's a huge problem across the country, but even more so in some states. Next slide. So what do we know about this? What do we know about what has caused this problem? What has contributed to it? And I think, despite what I said about federal policy earlier, there's been a lot of effort to kind of identify which attributes of nursing homes predicted which ones had cases and deaths. Even though, as I said, since most of them now have had cases, that's becoming a moot point. But still, we could hypothesize that some have done better than others and try to figure out why. And I've been combing through the data on this, and there have been at least 15 published analyses directly about the predictors of COVID-19 cases and deaths, and then some related literature. And they vary in terms of the data that's used. We now have national data that's a little bit problematic, but perhaps not in ways that really matter for trying to identify these predictors. Next slide. And so what can we conclude from looking across those studies? A few conclusions are really clear. One is that the strongest predictor of nursing home COVID-19 cases and deaths is simply the prevalence of the virus in the surrounding county, right? So nursing homes that are in hotspots are going to have cases and deaths. And that effect is just so much stronger than anything the nursing home actually does or any attributes of that nursing home. So this should give us, real food for thought about, any ideas about, protecting the vulnerable under a herd immunity program. Another conclusion we can make from that literature is that there's no meaningful relationship between the COVID-19 cases and deaths and the baseline quality of the nursing home, whether we look at the nursing home star ratings or prior infection control citations. So again, it's not so much what the nursing home is doing, it's where it's located and the prevalence of the virus around it. Next slide. Okay, and another conclusion we can make is that cases and deaths in nursing homes are highly correlated with race. I mean, the monotonicity of the numbers is really quite striking. And on this graph, you can see that, as the percentage of white residents in the facility goes up, the number of cases goes down pretty dramatically and monotonically. And same thing with deaths. And that's largely because race is highly correlated with where the virus is and a few other facility attributes like the size. That seems to be important. Next slide. Okay, as I said, a few nursing home attributes matter. It's not so much the quality, but larger facilities are definitely more at risk. Staffing can help a little bit, not surprisingly, but those effects are relatively small relative to prevalence of the virus. And you know, some of these things may emerge more, but I think this tells us that nursing homes are still in crisis mode. And you know, rather than punishing them for doing things wrong, what we should really be doing is assisting them. Next slide, please. Okay, a few implications to wrap up with. One is that the pandemic certainly represents an enormous challenge to all long-term care facilities, even the high-quality ones. Again, a little food for thought that, you know, maybe the attributes that lead to high quality may not be the same attributes that lead to handling a pandemic well. We were expecting nursing homes, for example, to put a lot of time and effort into procurement and trying to find PPE when none was available. And that's not necessarily, you know, the same skills that are needed for producing quality of care. We can certainly conclude that the pandemic is exacerbating racial disparities. And I didn't talk much about this because there's less research about it, but we know that, you know, the lockdowns that we have imposed on nursing homes have imposed their own costs in terms of social isolation. So, you know, even as we try to figure out how to prevent these cases and deaths, you know, the goal can't be zero risk of infection because we need to have some social interaction. Next slide. Okay, second set of implications. One, nothing much has changed. You know, unless we solve these problems of PPE and testing and get resources to the facilities in terms of, you know, upping their staffing and getting enough help with implementing what we now know to be best practices, like regular testing of staff, regular testing of residents, and cohorting of residents into COVID positive and COVID negative areas, and preferably cohorting of the staff as well, so they don't have to go back and forth, as well as PPE. In order to implement all of that, we need resources and we need staffing. And simply issuing guidelines is not going to really help the situation. Next slide. And so we're back to the policies. You know, like I said, there have been improvements in federal policy over the past few months. They've started shipping out PPE and testing supplies, but these are still sort of stopgap policies where, you know, there was like one shipment of PPE that was inconsistent in quality and then, you know, ran out. And they've sent out testing supplies to nursing homes, but there's no longer run plan to make sure that facilities have additional supplies when they run out of their additional shipment, and certainly no assistance with figuring out where to get those supplies or what the pricing will be. In the face of this lack of federal policy, you know, states have been sort of inconsistently successful in trying to step in. There's some things that they just can't do. They can't sort of fix the supply chain issue themselves, but some states have come out with programs like, you know, surge teams or sending in the National Guard or healthcare workers from other places to try to help nursing homes when they're in an outbreak phase. So, you know, that's helped somewhat, but states are limited in what they can do. And so then we look back at, you know, why is the whole industry in crisis? You know, nursing homes have basically been running in a daily state of crisis, you know, for decades. They're not really set up well. They're always putting out, you know, yesterday's fire. They're not really set up well to adapt to a challenge of this magnitude. And that kind of comes back to you know, how we pay for and deliver long-term care in this country, which needs some fundamental change. Next. Okay, so to wrap up, what can we do about all of this? It's a pretty dismal situation. In the short run, I think policymakers really need to fixate on the goal of preventing morbidity and mortality. So, trying to, sort of figure out who to blame for nursing home deaths and, worrying about helping organizations that might be for profit or that haven't done everything right in the past, I think is just sort of misguided, and it's going to lead to continued deaths in nursing homes. We have to sort of recognize it for the crisis that it is, and recognize that nursing homes during this pandemic may not be able to solve it on their own. And so I think federal policymakers, especially with the help of states, need to ensure that there will be PPE and rapid testing and assistance with staffing when needed, as long as the pandemic lasts, right? Not these one-time shipments, but as long as the pandemic lasts. In the long run, I think we need to really, reevaluate and improve how we pay for long-term care. Our system is very fragmented and chronically underfunded. And we need to rethink how we deliver long-term care in this country. These very large medicalized facilities that are, sort of perfect breeding grounds for a virus like this, those aren't really what any of us wants as we age. It's not a good model for many reasons. And so we have to think about, what the right replacement will be. And then finally, and these are all connected to each other. Finally, the people who work in nursing homes, it's sort of amazing they show up at all these days during this pandemic, but they basically make, minimum wage, are, below the poverty level, often endure pretty difficult working conditions and have no benefits. In the middle of a pandemic, many of them have no sick pay and no health insurance. And so I would argue that we're never going to improve long-term care in this country until we figure out how to support and improve the conditions of the long-term care workforce, which goes back to, this whole issue of how we're funding and delivering care. So I will leave it there. Thank you for your time.
Thank you, Tamara. You have a couple of questions and we have 30 seconds apiece. The first question comes in from Paul Nash. Could you please elaborate on the first conclusion about the rate in the local area? Are you saying it's impossible to shield patients or residents from the virus? Because this would have big implications for policies on lockdown.
Yeah, that's a great question. And I think it's one that, it's a little hard to know exactly where the right answer is. But yes, I think it's almost impossible to keep the virus out completely. And policies have to focus on identifying when a case comes in quickly and stopping it from spreading. And why is it so impossible to keep it out? Because staff are going in and out of the facilities daily, right? So what can we do, if the virus is highly prevalent and staff have a high chance of getting the virus and bringing it into the facility? What we really need is that regular, like semi-weekly testing of staff to try to identify it early and stop it from spreading. And then, of course, that goes hand in hand with the PPE.
And that feeds directly into the question from Holam Lai. Some research in other countries mentioned that nurse visits from hospitals to long-term care worsen the transmission. And is this the case also in the US?
Yes, I think any traffic in and out. So there's been research showing, for example, that facilities that have a lot of dialysis patients that have to go in and out to get their dialysis several times a week have higher infection rates. Any traffic in and out of the facility heightens the risk of infection. So what do we do about that? I don't think the answer is to just lock down facilities and try to prevent any traffic, because we know there are real costs to that, and there are some times when that's impossible. What we need to do is solve the testing problem so that we can identify those cases very early and prevent them from spreading throughout the rest of the facility.
Okay, super duper. And so we'll move. Thank you so much, Tamara. It was terrific, very informative, beautiful talk. David Meltzer is our next speaker, and he's coming to us from the University of Exeter and the University of Connecticut Center on Aging. Apparently he has bilocation. David, I welcome and thank you for giving us your time. Let's cue up the slides there.
Thanks very much for inviting me to do this talk on the epidemiology. Next slide, please. So the basics have already been touched on. It's obviously, we all know it's a respiratory virus mainly, spread through contact, airborne, with aerosols forming in confined spaces. The international meta-analysis suggests that the incubation period is about 4 to 6 days, with a 95% confidence, an upper limit of about 12.5 days. As has been mentioned, it kind of stands out as a little bit different in that the great majority of test positives seem to be asymptomatic. So in a random population sample taken in the UK recently, 76% of those testing positive turned out to have no symptoms at all, no specific symptoms. And only about 14% had the classical symptoms of a new fever, new cough, new loss of smell or taste. So a huge variation in presentation, and older patients tend to present in less typical ways, so it can be quite difficult to diagnose them. The virus RNA sequence has changed very little since it first appeared. No substantial differences as far as we can tell. So that the enormous differences we are seeing from place to place, community to community, and individual to individual are due to the social factors and the host vulnerability factors. And I'll focus mainly on the host vulnerability factors. Thanks. Can I have the next slide? So there's been a lot of press coverage of the nature of the patients being admitted to hospital. These are data on 88,000 hospitalized patients from a number of countries. And as you can see, in terms of numbers, the older group is very dominant. The blue bars are the numbers dying. And as you can see, the proportions dying, especially in the older men and also in the older women, are very, very substantial internationally. But we should note that they're pretty big numbers of 50-year-olds, 40-year-olds, 30-year-olds, and even younger who are being admitted to hospital and are dying. So it's not uniquely an aging problem, but it is dominated by older ages. Thanks. Next slide. A lot of focus early on the top conditions that were being seen. So there was much discussion of hypertension, heart disease, and diabetes being very common in the patients. This is from the same international collaboration. But actually, the big problem with this is that these things are pretty common in older people anyway. So next slide. What we did is we have a big cohort, 500,000 strong cohort in the UK Biobank, covering England, Scotland, and Wales. And we were able to look at the 269,000 people in that, age 65 and over, and look at the risk of being admitted to hospital and testing positive. We published this in the Journals of Gerontology a few months ago. This was early in the epidemic. Very clear increase in hospitalization rates with age, although from 65 to 70 was our control group. The risks aren't quite as big as people say when you take a population view. Male sex is very much a risk factor, almost doubling the risk of hospitalization in this cohort. And as we've heard, minority ethnicity, Black British, South Asian ethnicity, and other minority ethnicities are more than doubling the risks of being admitted to hospital with serious infections. Next slide. So in terms of those diseases, you'll remember that coronary heart disease is very common in the patients, but actually, if you look at the background population from whom the patients came, as we're able to do in UK Biobank, there's no excess risk for people with heart disease at all. The hazard ratio, odds ratio is pretty well near 1 and not significant. The biggest risk factor was actually dementia, for many of the reasons we've just heard, that people, especially people in nursing homes, are at very high risk. But of course, some of those risk factors also apply to home care. We're seeing odds ratios of 3.5 for hospitalization and even bigger for death with dementia. Then diabetes is definitely a risk factor. Hypertension isn't much of a risk factor. We found some evidence for atrial fibrillation, heart fluttering, abnormal rhythm of the heart as a risk factor, which would fit with perfusion, heart output being an important thing. And that's not attracted a lot of attention so far in assessing individuals, but it's very clear that there are specific diseases, specific pre-existing diagnoses that increase risks of being admitted to hospital with COVID-19 in this older group, the 65 and overs. Thanks. Next slide. So genetic epidemiology, looking at the inherited genes that seem to be associated with more severe COVID infection, has the potential to tell us something about the biology in humans, which no other kind of evidence, or it's very difficult to get near causal evidence in any other way in humans. The first genome-wide association study, GWAS, was of respiratory failure patients admitted to hospital in Italy and Spain. This was published actually in June, but appeared last week in the NEJM. 835 patients, 1,255 controls. The controls were drawn from a number of different data sets. We'll come back to that. And one of they got two strong indications. One, a gene called SLC6A20, which is very interesting because it interacts with the ACE2 receptor, which is the protein on the surface of endothelial cells in the lung and in other cells throughout the body that the virus binds to in order to enter the cell and infect it. So that seemed pretty exciting, but actually the genetic variant is associated with a whole bunch of genes, and work is still going on to sort out exactly which variants, which pathway is important. There are also a number of immune genes in that pathway. And then that paper originally got a huge amount of attention for blood group, ABO blood group, but actually the replication of that's been very limited, and basically nobody's been able to replicate that. And the guess is that that was because they used blood transfusion controls, and as I'm sure you know, blood transfusion services tend to favor certain blood groups over others, which would have introduced a bias into their controls. So quite an interesting technical problem there, but it doesn't look in the more recent studies that ABO is actually a risk factor. Next slide. The second GWAS that's come along was of critically ill patients. So these are people in intensive care units. And they're pretty interesting people because firstly, they're fairly rare. And secondly, they respond to steroids. There was a trial in the UK showing that they respond to steroid treatment when people with lesser severities of this disease don't. It's actually dangerous to give steroids early on in the disease. It's only in these very severely affected people. A group has the genetics of mortality and critical care group has pulled together data on 2,244 patients, so quite a lot bigger than the first study. And they've very much replicated that SLC6 marker I mentioned. But what's pretty exciting about this is most of you will probably know that GWAS results are usually tiny odds ratios. 10%, 20% increase in risk is regarded as big in most GWASs. Here, that top hit actually doubles the risk of percent of the population carry that variant that doubles their risk of ending up as a critically ill COVID patient. They also found a bunch of signals for innate antiviral defenses. So this is a gene cluster, the OAS1, 2, and 3 gene cluster, which produces a restriction enzyme which limits the virus's activities directly. And it's pretty exciting that this is a druggable target, along with the second one, which is an interferon receptor. And so we're beginning to get potentially druggable targets from the genetic epidemiology. Next slide. And then I should have mentioned that that second study is on preprint at medRxiv. The third study, unfortunately, is not published in a conventional way. It's part of a very big international collaboration on the host genetics of COVID, but there have been public meetings that are accessible at the website there. And they reported their results in October for 17,000 hospitalized patients and 1.3 million controls. And they replicated the ones we've talked about. And they also found pretty convincing evidence and replication of variants involved in lung epithelial cell injury resolution. So this is the lining of the lung and its ability to heal itself. So one of the variants has previously been linked to fibrosis in the lung, interstitial lung disease, and the other has been linked to lung cancer. So it looks as if you're unlucky enough to have suboptimal injury resolution in the lung, you might be at higher risk. And also they've extended the immune response signal with a number of autoimmune variants. So TYK2 has previously been linked to a bunch of autoimmune diseases, rheumatoid arthritis, SLE, autoimmune hypothyroidism. And the HLA region, this is the classical autoimmune region. So we're seeing another group of patients with autoimmune vulnerabilities at higher risk of hospitalization. And the last new one, CCR2, is a monocyte and macrophage attractant chemical. And one of the interesting features is that the lungs of these people tend to be full of macrophages. They also looked at gene scores for body mass index and smoking, and both were pretty strongly associated. So those do look as if they are independent causal risk factors. Next slide. Those GWASs look for common variants. The ApoE44 variant is pretty rare. This is the one that increases the risk of Alzheimer's disease by over 10-fold. We looked at that in the UK biobank study, and we published a couple of studies on that and the mortality. That's Ku et al. From Connecticut, in which we showed very strong associations for this small group of people with the double E4 variant that's been studied so much in aging. Thanks. Next slide. So in conclusion, the epidemiology is showing a number of vulnerable groups. males, ethnic minorities, and a number of specific comorbidities. We're getting very exciting signals from the GWAS around immune response, lung repair, and viral control genes, as well as ApoE, E4, E4. And I guess the big conclusions in broad terms are there's very little support for the crude age-based targeting of older populations that we've seen in a number of countries. And also, all this epidemiology is fine, but the dominant risk factor, age, I've put a graph from the case fatality rate from 3 different countries from May 2020. And as you can see, very different countries, very different background levels of COVID, Italy, Japan, and Spain, but almost identical age proportions dying. And we still see that age is a dominant factor and largely unexplained. Obviously, comorbidities explain it to some extent, but largely unexplained. So thanks very much. The biobank work was done by people in Exeter and Connecticut, and have to acknowledge their input. Thanks.
Thanks a million, David. That was an amazing talk. I'm super interested in that DPP9 because it's a cell surface receptor, right? Does it interact in any way with the ACE2 receptors? Is it in the same geographical location?
Pass. I'd be bluffing if I made up an answer on that one. I'll check.
Well, here's a better question. I saw that there is a profound increase in the risk for patients with dementia. Do you know what that's about? They really sat outside on your graph for the people who ended up becoming hospitalized.
Right. So I think part of it is because people in care settings are, there are high rates of dementia in people in nursing homes. And secondly, it does look as if ApoE44, E4, the Alzheimer's high risk gene, is a risk factor. We actually had no people with actual dementia. It was just the genetic variant itself, the mutation itself, which of course is also shortens life generally. That was very strongly associated with risk. So it might be both the care factors we heard about in the last talk, but also some intrinsic vulnerability in those pathways that cause Alzheimer's disease.
Mm-hmm. And we have a question from Terry Lewinson. Interesting that asthma did not have a stronger relationship.
Right, I should have mentioned that asthma for people on steroids, so the severe end of asthma, is a risk factor. Their risks are raised. But if you, for patients with just ordinary asthma, not on oral steroids, taking steroid tablets, they can be on inhaled steroids, that's fine. They don't seem to be at an increased risk.
Okay, superb. Well, thank you so much. Yeah, and you got a thank you back from Terry. And so I think we'll move on to our next speaker, Janko Nikolic-Zugic, who's coming to us from the University of Arizona Center on Aging. Janko, take it away. You might have to unmute, maybe.
All right, can you hear me now?
We can indeed, thanks.
Lovely. Thank you for the invitation. We can immediately move to the next slide. I'm going to be talking a little bit about, and actually, if you can click again, I think there's unfortunately some animation here. Those were the people that I wanted to credit for doing the work. The top part, my lab, and the bottom part, various collaborators who have helped mightily and critically in what I'm going to be talking about. So in the next slide, actually, we can go 2 slides ahead. We're going to be discussing a little bit about the virus. We've heard a lot about it from the epidemiology standpoint. I just wanted to touch upon of what makes this virus virologically different from its cousins. And if you can click this one more time, we're going to have some more animation coming up. In addition to being able to do a lot of different things that SARS coronaviruses normally do, this virus has mutated its binding site so that it binds much better and gets much more efficiently inside the human cells. So you see here this spike protein called S on the very top of the spike protein. There is a part of the virus that actually the virus uses to attach it to our own cells and gain entry inside the cells. As a reminder, viruses cannot replicate outside the cells. They have to be inside the cell in order to turn the cell into a factory that's going to make more viruses. And that's how they spread around. That's how they make us sick. That's how they eventually can also kill us. And this is this very part that that binds to the human angiotensin enzyme 2, converting enzyme 2, has changed so that it now can bind about 15 to 20-fold better and more strongly than its original cousin SARS-1. So this virus has just a higher affinity for our cells. You have asked, and David has posted all of these GWAS studies. This is not, ACE is not the only molecule that this virus will use. There's a whole slew of different other molecules that the virus might use as other cofactors for entry. DMPP that you have mentioned, and as well as Timpress and many other surface molecules can be used in this process to do it, but less efficiently. And what determines this rate of interaction really is the affinity of the spike protein receptor binding domain, or what's abbreviated here as RBD, binds really, really well to human ACE2. And then the other thing is that, in addition to being a respiratory infection that makes this upfront a nightmare for spreading between humans, this virus persists in the environment and seems to be much hardier than its cousins SARS-1 or MERS. And of course, the other thing you have heard from both of the previous speakers, the virus is shed before we get sick, and so that is a big problem. Even on top of that, the asymptomatic transmission adds another kink and another twist. So this is all what this virus does to spread better, to gain better access, to infect more people. So it's very, very good at that. Next slide, please. But what the disease is like, and this is again just a quick review. This virus belongs to the coronaviruses that have shown a very strong propensity to do what this virus did, which is to change and then jump hosts. Coronaviruses are very widely spread between different species in the animal kingdom, and when these species are in close proximity, the virus will change and will try to gain entry into the new host. It doesn't change like crazy. It's not like HIV. It's not like the hepatitis C virus. It's not a super rapidly mutating virus, but it is an RNA virus, which means that the primary mode of its evasion is to change itself to avoid the immune system and to gain entry into the new hosts. So the disease COVID-19 is characterized by all of these symptoms that are very well known by now: fever, cough, fatigue, shortness of breath, acute respiratory distress syndrome, and in some people, cytokine storm. So we do not understand yet, as David had mentioned, why some people get very vulnerable. It's very clear that younger individuals can get very, very vulnerable as well. Next click, please. But this is something that you have already heard, and this is just the situation in Arizona that says that people already between 45 and 54 are about 3 times more likely to die than those between 21 and 45. And then this mortality goes up another 10-fold between 55 and 64, and then it culminates at very, very high rates, those over 65. So very pronounced, by far the largest risk factor is your age. So let's discuss a little bit how and why and what would be the problems with your age. So there are many issues that of course change with aging, and the one that we're focused is how the immune system reacts. And you've heard from David that even the initial sensing of the virus is problematic, that these innate sensors in people that are severely sick or die tend to have problems in the early detection of the virus, and particularly along the type 1 interferon, which is the primary sensing mechanism for most viruses. But this slide here shows how our white blood cells coordinate an immune response after that initial detection of the virus. And so here on the left-hand side in the very top corner, there is an interaction between a dendritic cell and a helper T cell, or a T cell in general. So this dendritic cell is a sentinel cell that has to detect the virus, pick it up and bring it into our lymph nodes typically, or other lymphoid tissue. That's where these dendritic cells will activate our T cells. Our T cells are in charge of recognizing the virus, secreting various cytokines, which are chemicals that can stop viral replication. One flavor of them actually will go and kill directly the infected cell, because the strategy here is like, let's eliminate the factory that makes the virus, rather than wait around for this factory to make thousands of more copies of the virus, burst and release them around and perpetrate the infection. That's what some other T cells do. On the other hand, in the other side of this slide, you've got purple colored B cells. They're the ones that make antibodies. They get activated independently. These two types of cells then get together in the middle of the slide. You can see a red T cell and a purple B cell interacting. This will help B cells make better antibodies and stronger antibodies and everything else. And this is what will eventually put a stop to the viral infection, this coordinated production of antibodies and antiviral T cells. Unfortunately, most of these components, and this has to be a very highly coordinated and very speedy interaction. You need to have proper and quick communication between all these cell types. They also need to travel to the right site to meet each other. And all of these components of this process are compromised with aging. So let's go to the next slide and look at how this happens in another view. So here in the top portion, you have the situation in the adult. Again, in the upper left corner, you've got these little pieces which are denoting the virus. And this virus is picked up by the dendritic cell, which is this little cell that then becomes a bigger cell. I don't know whether you can see my cursor at all. I don't think so that you can. But this dendritic cell, which is this cell in the center of the slide, is like now bigger, and it's colored brightly red. And that cell comes to talk to and communicate with the T cells that are in the lymph node. And we have quite a bit of these cells that we call naive T cells. Amongst them, this dendritic cell needs to secrete to find the ones that are specific for the coronavirus, for the SARS coronavirus. And only one in about 100,000 is going to be specific for it. But by the process of intense trafficking and attraction to particular microenvironments in the lymph node, you can actually get these cells to be close to one another. And then as a consequence, in the upper part here, you will have now major proliferation. A lot of these cells have been made. You can see a whole slew of them. And they also have they're very well armed to fight the virus. They have all of the antiviral chemicals in them. What happens in aging is shown in the bottom part of the slide. The initial detection of the virus that is supposed to happen by the dendritic cell is not very efficient. And maybe this sensing through type 1 interferon is one of the problems. Most certainly is in some people genetically. But in older people, this is actually slow just as a consequence of aging. So this dendritic cell in the center is not nearly as big as the one in the adult, meaning it's not as activated. It's not making as many of the goodies that need to tell the T cells to get activated. But you can see in the left bottom corner that there's a small pool of T cells now, and that is another problem. We have lost a lot of our naive T cells that we used to have in youth, and that is because there's a problem of their production. So as a consequence, now in the right-hand side of the slide, in the bottom part, we do not have nearly as many defenders as we need to. Not enough T cells have been made that are proliferating and that are now armed and dangerous and can kill the virus. And so that's one of the big problems. So in the next slide illustrates the problem in production. The reason why we have fewer of these naive T cells is that instead of what's shown in the left side, there's a big thymus. The thymus is a gland that sits above our heart and underneath our chest bone that makes T cells. In youth, it's making a lot of T cells. By the time we hit puberty, that declines by about tenfold. By the time we get into our 50s, it declines by another tenfold, so it's down to only 1% of what used to be made. So we have a real problem with production, and as a consequence of that, we no longer have enough naive T cells to respond. If you can do the next click, please. There's also a problem in the maintenance of these cells, but we're not going to delve in that too much. That's a problem of changes that happen in secondary lymphoid organs, primarily in the lymph nodes. And another click that summarizes all of this. So essentially, new T cell production diminishes early. Peripheral maintenance works for decades very well, but in the last third of life, we lose our naive T cells, which are supposed to defend us against new infections that we have never seen before. And so that is one of the key problems that we're facing with aging. We don't have enough cells to respond. Their coordination of the response and maintenance is not quite as good. And the signals that they get in the course of activation are suboptimal. So all of these, even if they're reduced by a little bit, by a 10% or 15% factor, because they're running in sequence, in the end, you have a result of many, many fewer defenders that are there and ready to respond. The good news is that we can manipulate this process and make it better, but that's a story for another time. So let's go back to our coronavirus in the next slide and talk about what we wanted to do and what we wanted to find out, us and the rest of the world. So in late February, early March 2020, we wanted to simply detect the immunity to the virus and figure out what parts of the virus may be targeted by the immune response in adults as opposed to the old, what immune protection versus immune evasion may be happening, what is the duration of immunity, and most importantly, the bottom question, is there an immunological basis to vulnerability to this particular virus in older adults? So next slide. So we decided to put together an antibody test, and the moment that we started doing this, we got interviewed for a local, actually not local, but Arizona-wide, Arizona Republic, which comes out in Phoenix. And if you can click it one more time, we told them very clearly here, as you can see in this quote, that our tests will not be used to conduct widespread testing in the population. We were making it just for our research only. Fast forward less than 2 weeks, next slide, there was this announcement that came from our governor saying that this test that we were putting together will be testing 250,000 people. So we said, oops, I think we're in the hook for something a lot more than what we thought about. So we had to speed up with the production of this test. Next slide. These are my collaborators who helped a great deal with putting of this test together, Deepta Bhattacharya, Ryan Sprisler. Next slide. The test that we have generated, you can keep clicking until everything comes up. Don't worry about it. Initially, it was a single-step test. We figured out that this was not specific enough. We had to add another piece of the virus to make it much more specific, because in our population, we had very few people who were infected at that time. Our false positive rate is very, very low because of this. And we can show that everybody here that's colored red is also producing neutralizing antibodies. So we had a very high correlation between people making high levels of antibodies overall and producing good neutralizing titers, which means that these antibodies essentially can block the entry of the virus inside the cell. We tested more than 30,000 people now, and we know now that seroprevalence is roughly about 3 to 5% in Arizona. Next slide. But the really important question was, there were reports coming out even in as late as July that we might not have lasting immunity against this virus because antibodies are disappearing. We were very perplexed and very, very upset at this, because this really reflected fundamental lack of understanding and knowledge of immunology and virology. You can move to the next slide. We know that antibodies in general last very, very long. these are half-lives in the box that you can see in men and women in years of how long antibodies last. And you can see that in some cases there's no decay, meaning that they last indefinitely. In other cases, they were calculated to be for measles in the bottom of the figure, for instance, for males, that their half-life would be 370 years, meaning that our immune system would cover us for about 15 to 20 lifetimes, if not more. So having antibodies that disappear rapidly didn't really make a lot of sense. Next slide. And we believe that this was just an error of interpretation, because normally antibody is produced in the first wave just to give us some protection over the first 2 to 4 weeks. And those are short-lived cells that make them, and the antibodies are not very good. This is depicted by this first violet peak that you see here. That response goes down relatively quickly. There's a decay phase of it. You can see that it goes up and then it goes down. But what's depicted here in green are really long-lived cells that come from the germinal center. It's depicted here as the germinal center response, and they make very high-quality antibodies. And these antibodies are really not likely to go away very quickly. So this, extrapolating from this initial fall in antibody titers to saying that in 2, 3, 4, 5, or even 8 weeks we will have no longer any antibodies, was absolutely wrong. And next slide. We could actually show that in our own studies. Here you can see that we have been running antibody tests for one part of the coronavirus in the left side, and the blue line is the cumulative one that for both people with mild disease or even asymptomatics, these antibodies were maintained very well over 200, close to 250 days for the other part of the virus as well. And next slide, please. Most importantly, when we look at neutralizing titers, these seem to be very stable here. Again, for both types, if you look at the blue line, it is up, it goes down a little bit, but then it's actually running fairly flat for a while. So based on what we know about coronaviruses, we really expect that there's going to be a couple of years immunity at the least. And then for you know, we know for SARS coronavirus that many people still have immunity 17 years out. There's a lot of debate whether this is going to be true for everybody or for people who got even a little bit of virus, whether the dose is going to play into that. Those are the questions that we cannot answer right now, but there's no way that we're going to be left with no immunity after several weeks only. Next slide, please. Very quickly, we have also done some single cell RNA-seq to look at the remodeling. And you can see here, this is week 1 on the left and week 2 on the right. And you can see that just by the whole bunch of cell types is disappearing relative between week 1 and week 2. Like for instance, the big blob of orange cells marked CD4s is very depleted. Many other cell types as well. The B cells are down, the monocytes are down. So we're still trying to understand what's going on. I'll give you a quick summary or preliminary data on the next slide. We know that there is correlation with disease severity in the literature. It seems to be stronger than that one with age for different types of immune responses. But the problem is that studies of that are well controlled for age are very rare. The interpretation is still confounded by our unknown viral loads. We don't know who got how much virus, so we cannot stratify them by that. But what is very clear is that there's severe remodeling of immune cells in the blood with severe disease, and definitely not in the mild disease. We're starting to disentangle signatures of innate and adaptive cell transcriptomes. This is starting to provide initial clues at what's going on. And there's a lot of things to do here, and I will stop now and take any questions that you might have.
Thank you so much, Janko. You have 15 seconds remaining in your entirety of allotted time. There was one question that came in about thyroid functioning and targeting improvements in thyroid as a means to boost immunity in older age. Can you answer it in 7 seconds or less?
Not much is known about it, and I don't think that I can comment cogently on that.
Yeah, Well, we hopefully will have time at the very end for a panel discussion, and I encourage people who are attending to please load some questions into the question box, and we'll be able to deal with them at the very end. Thank you, Janko. That was terrific talk and really exciting data. Okay, so we'll move on to our next speaker, who is Brian Kennedy, who comes to us from National University System Singapore School of Medicine and also the Buck Institute for Research and Aging in California. Brian, welcome and thanks for your time.
Thank you. Thanks for inviting me. I have to say that I'm coming from this way more from the aging side than from the COVID-19 side. So I'll try to say some enlightening things, but I think it might be better if we make sure Janko's speaker is muted so he can't, otherwise he's going to sound like Trump jumping in and telling me all the crazy things I'm saying. So, but I'll try. So I want to start with, if you could go to the next slide. And I want to start from the aging side and then I'll come to the virus side. And we've been making this case in the aging field for a long time now that we treat chronic diseases of aging as individual diseases and don't consider the connectedness between them. And in fact, if we move over to the right side of this slide, what we really don't consider is that aging is the biggest risk factor for a wide range of different chronic diseases. And I think that the idea then, of course, is that if we can target aging, we can protect ourselves from a whole range of different diseases simultaneously. And I think one thing this year has hammered into my brain is that we were not thinking about this in a complete way when we were using these slides over the last 10 or 15 years. If I could have the next slide. As you've already seen, the risk of mortality from COVID-19, the age is the biggest risk factor. And this is not particular, this is on the left in this little box, so it's really hard to see. If you go over to the right, though, this was probably not that surprising because for a lot of infectious disease, aging is the biggest risk factor for mortality. This is data for influenza on the right. And so if you go to the next slide, what we really should be saying is aging is not just a risk for chronic diseases, but it's a risk for the severity of acute diseases, infectious diseases. And it's also, of course, a risk factor for functional decline. And so I think that makes it even more relevant that we think about how to target aging pathways. Now, I will point out that of the $4 billion allocated to COVID-19 research by NIH, as far as I can tell, none of it went to the National Institute of Aging. And so there's still, I think, is an underappreciation of this concept that we need to understand what it is about aging that contributes both to chronic and infectious diseases. And by the way, if we can figure that out and do something about it, then we'll have people that are more protected from the next pandemic that comes along as well, which is very likely to affect older people. If I can go to the next slide. Of course, what does it mean to do interventions to slow aging? And there's really three kinds of interventions that have been considered. One is lifestyle interventions, which I would put in this dark red circle here. These are already in the public. There's already mechanisms there's pretty good data for, exercise being a great example that slows aging and protects people from various diseases. I work in this intermediate space here, which is drugs and supplements that we're trying to identify that slow aging. And then, there are futuristic ideas like stem cells and gene therapy that I'm not going to go into today. So I think that we're at this point in the aging field where we can now think about how do we validate the idea that we can either slow or reverse components of aging. So if you go to the next slide, these are all small molecules that I work on in my lab. And I'm going to show you just a little bit of data on alpha-ketoglutarate. But I want to point out that all these other compounds in gray here are already being considered for their ability to treat or protect you from COVID-19. And I'm just going to go over a couple of those because I think it's intriguing. I didn't circle NSAIDs, although there's a lot of data on NSAIDs in COVID-19, or a lot of speculation as well. But I'll start with alpha-ketoglutarate. Next slide. I'm not going to go into this too much because this is not the point of this talk, but we just published a paper in Cell Metabolism showing that alpha-ketoglutarate in mice compresses morbidity. So it has a very modest effect on lifespan extension. It's about 5 to 10%. I'm only showing you females. There's a little bit lesser effect in males. But what it really does is it compresses morbidity by about, reduces frailty by about 50% in these animals. And I'm not going to go through the meaning of this slide, just to tell you that the red curve here is the AKG treated animals, and essentially they're largely healthy until the very end of life, and then they keel over and die. So they have a very long health span. Next slide. And we've been looking at what the mechanisms are, and one of the mechanisms we discovered may be relevant to COVID, so that's why I wanted to talk about it. And it comes back to this fact that people have been taking alpha-ketoglutarate forever to try to enhance athletic performance, enhance endurance, enhance muscle building, wound healing, and nobody really knows why alpha-ketoglutarate might be beneficial. And the clue for us was that even though we were giving this to animals and it was having an interesting effect on aging, a lot of cells don't take up alpha-ketoglutarate. And then, so we started looking for what cells in the body actually do take it up. Next slide. And it turns out that red blood cells have specific mechanisms to take up alpha-ketoglutarate. And there's a reason for that, because they don't have normal mitochondrial function. So they need to make a lot of molecules very differently than your other cells. And alpha-ketoglutarate is the main limiting reagent for production of glutathione in red blood cells, which glutathione is an antioxidant defense mechanism that protects hemoglobin and a lot of other molecules in red blood cells and improves red blood cell function. So next slide. So what we can show is that either in cell culture on the left, into red blood cells, if we add AKG, we get a big boost in glutathione production relatively rapidly. And if we treat animals with AKG, we can actually see their red blood cells have more glutathione. So we now have this model where glutathione, we know glutathione decreases with aging in red blood cells. And unlike other cells, red blood cells take up AKG and make glutathione with it. And they're also particularly sensitive to oxidative stress because they carry a lot of oxygen around on hemoglobin. So we think restoring glutathione is likely to increase red blood cell function and oxygen delivery, which is good for building muscle if you're exercising. Next slide. It may be good for aging too. And it also may be good for COVID-19 because reduced oxygen saturation and acute respiratory distress is what generally, if you look on this graph here, is a major predictor of mortality in hospital patients. And the primary defect, of course, is not in the red blood cells, but if you can improve the function of the red blood cells, you may be able to maintain oxygen levels higher in tissues for people in acute conditions. So we're interested in whether that might be a thing to do clinical testing on. And when we got in lockdown, I also told my postdocs to go think about something since they can't work in the lab and come back with an idea for a review or a new experiment. And so Xiaoping, next slide, took this very seriously. And he's come up, oh, sorry, I missed the whole second, I missed the second component of the talk. I'll come back to that. And I do need the next slide. I just need to, sorry, it's 1:15 in the morning here, I'm trying. So I just wanted to go through the other molecules that slow aging. And all of them have been suggested that they might affect COVID-19. It's already been shown in 2 clinical trials that using rapalogs can protect individuals from infectious disease with only 6 weeks treatment and with the protection lasting up to 9 months after that treatment stopped. There was a phase 3 trial by this company that did not work, to be fair, but they also did not use a rapalog, which is a specific TOR inhibitor that's been linked to aging. So we don't know why that is, but so rapamycin has been speculated to be beneficial. Next slide. There's been a lot of talk about metformin and some papers and meta-analysis. The conclusion from one of the recent ones was that metformin significantly reduced odds of mortality. This may be linked to the risk of diabetes. Another study came out and said it didn't reduce mortality, but it did protect against cardiovascular complications, although it had higher acidosis. So there's a lot of debate around metformin. Next slide. There's NAD precursors have been linked to COVID-19 because apparently the SARS virus, this virus infection leads to deregulation of enzymes involved in NAD biosynthesis. It actually upregulates these non-canonical PARP enzymes that consume NAD, and that may be a protective mechanism for the virus. And restoring NAD levels, at least in cells, can inhibit some kinds of viral production. So there's speculation there. And so next slide. So I guess there's two ideas right now. Are there really connections between pathways linked to aging and viral defense mechanisms? And that's why all of these longevity molecules may have some beneficial role. It could be related to innate immunity or anti-inflammatory effects of a lot of these molecules. Or the second idea, which I think is equally possible right now, is when all you have to do is drive in a nail, everything looks like a hammer. And it may be that everybody's just trying to fit their favorite molecules into a potential treatment for COVID. And I think that a lot more research needs to be done to see if there really is a link to longevity molecules and viral defense mechanisms. But there's some intriguing ideas out there. I'll leave it at that. And then finally, I want to go back to my postdoc in the next slide, who has been looking at nitric oxide levels. So you heard a lot about adaptive immunity from Janko. But there's also cell autonomous immunity, and through interferons, cells can protect themselves as a first line of defense, endothelial cells, for instance, from viral infection. But it's also true that nitric oxide is protective from viruses. And the interesting thing about nitric oxide is it declines rapidly with age in multiple studies. Next slide. And there's two different enzymes that produce nitric oxide. There's endothelial nitric oxide in endothelial cells. There's also an inducible form. And most people tend to ignore the endothelial nitric oxide because it doesn't produce nearly as much as the inducible form. But it's been shown that in endothelial cells, when you have a lot of inflammatory stress through the bradykinin pathway, you can superactivate eNOS, and that actually produces a very high output of nitric oxide. So in an inflammatory condition like COVID, you might have very high eNOS levels producing pretty high levels of nitric oxide. Next slide. And nitric oxide has been shown to have many different effects on inhibiting viruses, including inhibiting viral replication. This is a cell culture experiment where adding a nitric oxide donor increases nitric oxide levels and inhibits influenza replication. Nothing to my knowledge has been done yet on COVID-19. Next slide. And so we put this together in a review just as a concept paper and put it in because don't really have an easy way to look at it. And this is one of the most interesting comments I've seen from a reviewer, helpful comments. So if you happen to be in the audience, thank you. They said that you may not know this, but the NOS3, which encodes the endothelial enzyme, has a lot of different mutations in it and that are very common in the population. So there are many different haplotypes, and those are associated with different levels of activity of the enzyme. And so if you could look at different populations around the world, you might be able to see a link between mortality and wild type levels of this enzyme. And in fact, that's exactly what we see. If you look in countries that have a very high death rate per million, they tend to have less than 50% of the people have the wild type haplotype for this enzyme, where if you look in regions that have very low death rates, they tend to have a very high percentage of this haplotype. Now, these studies, this R squared value is pretty striking, but these studies, there are lots of complications behind them. So I'm not trying to over-represent this data, but I do think it's very intriguing that we should be looking at eNOS as a potential mechanism for the loss of nitric oxide production as a potential mechanism with aging that may contribute to some of the sensitivity to this virus. Next slide. And of course, it's also intriguing because there are lots of different ways to drive nitric oxide levels up in cells. And they're everything from folic acid to estrogen, DHEA, statins. Some of these drugs have other complications that may make them not useful in this context. Here's another aging molecule, resveratrol, that's reported to do that. And so we're not without strategies to alter levels of eNOS activity. So it may be worth considering some of these approaches as well. when we think about things in the clinic. And another aging molecule, vitamin D, that we work on is also a regulator of endothelial nitric oxide synthase. So I think that we're looking a little bit more into this idea that nitric oxide could be one of the reasons that there's a differential response among aging population to infection by COVID-19. Next slide. And I think that's where I want to stop. I just want to thank mainly Guangzhou Ping in the lab, who is doing the AKG studies, but also when he couldn't do that, spent a lot of time reading about nitric oxide and came up with this concept as well. And hopefully that'll be published soon. And I'm happy to take questions.
Thanks a million, Brian. That was terrific. I'm actually a bit curious about the nitric oxide. Isn't this, massively studied in the context of vascular health? So there are all kinds of kits and caboodles that could be brought to bear and ask if they have any penetrance in COVID susceptibility.
Yeah, as far as I know, I think that's true. I'm just catching up to speed on this myself. We should have had my postdoc do this talk. But yeah, I think it's certainly linked to that. And those polymorphisms in the enzyme are also linked to cardiovascular risk. And that may explain also why certain Asian populations are less prone to cardiovascular events than Western populations. There's already literature on that. So it's pretty intriguing.
No, the biology is just fascinating. And I think it's pretty clear to everybody in attendance that, without looking at aging in addition to COVID, we're really not going to be able to make the kind of advances we need given the prevalence of the disease. So we have some questions, open questions now. Please use the question box, everybody, and please join me also in thanking all of our speakers who are absolutely terrific. I just want to get that out there ahead of time in case we get automatically shut down. I want one announcement to make is that the session is recorded, so you'll be able to go in and take another look at these presentations. There's so much information, a second viewing wouldn't do any harm. With that, I think I'll just go to the questions in the box. So we have one for Yanko from Rebecca Reed. Do they have preliminary data and could you speculate about latent virus seropositivity and vulnerability to COVID in younger people?
Yes, so am I unmuted? Can you hear me?
Yes, but we'll keep it nice and quick, right?
Sure. So the protracted virus positivity dominantly comes from shedding of cells that used to be infected with the virus, and they're now, turning over and dying off in the nose and in the mouth. It's really not a sign of active disease, and people with antibodies are not going to have any problem, fending off the infection. They're not going to be infectious. Second part of the question was?
Well, where are we? Younger people.
Yeah. I mean, so younger people, again, are not bulletproof by any means, and that's been one of the biggest and most dangerous misconceptions about this virus. We really have to remain careful. Many people from full health and very young age have ended up very severely ill, as well as dead, unfortunately. So do not let your guard down. Quick comment on Brian's stuff and nitric oxide. Endothelial pathology and pathogenesis for this broad spread of the virus across different organs and hurting different organs is one of the leading theories. It'd be really interesting to test that with your nitric oxide stuff.
Thanks.
We have a question from Jamie Justice. I think this is for Brian. Curious if we're looking at the right outcomes and strategies for these aging biology targeted interventions. Perhaps these host-directed therapies may not be terribly protective in the short term for acute viral load or hospital outcomes. How would you propose evaluating aging molecules in the long-term health effects after SARS-CoV-2 exposure?
Yeah, and some of these things were surprising to me too. We're thinking of intervening in aging more as a preventative strategy. In other words, if we can alter your biologic age and protect you from disease, then you will be protected prior to getting infected from the virus, which is probably the best strategy in terms of targeting a risk factor like aging. I kind of view the AKG thing as just unique overlap with something that might be beneficial in terms of improving oxygen carrying capacity. But you know, I just wanted to point out that some of these more acute potential benefits of these longevity molecules, and I honestly don't know what that means either. We're thinking more of long-term prevention.
Brian, you've one more question from Mary Murphy. What supplements are you taking in the COVID era? AKG,
The only thing I'm taking is the product from the company that sponsored the research, which is Rejuvent. So it has AKG and low-dose vitamin A.
Okay, cool. So I can open things up now for discussion among panelists. We have a few moments only, and I don't want to, you know, derail any levels of discussions. David, you mentioned a thing about steroids early in the condition being detrimental. Can you elaborate a little bit on that?
So there was a randomized controlled trial in hospital patients, and what it showed was just dramatically better outcomes in people in intensive care units in acute respiratory distress, but actually a bit of a trend in the other direction for the less severe patients. So it looks as if high-dose steroids anyway is only helpful in that phase of very severe disease where there's, you know, growing respiratory failure, and I understand a kind of immune storm that causes a kind of runaway immune response. So yeah, there's a lot of interest as to why it should be so beneficial late in the disease, but actually trending harmful earlier on.
And that makes perfect sense in terms of the kinetics of the immune response. You know, you do not want to harm the initial ramp up if it is properly regulated. Obviously, once it gets out of hand, you want to dampen it with steroids. Steroids will do both things.
Yeah. I think, David, Tamara, if you have a chance to comment, you know, I think this is one of the things, particularly in the elderly, that present with comorbidities and often have polypharmacy. And so, you know, do you know what steps are being taken to, you know, introduce those caveats when people are coming in for critical care? You might be muted. Oh, there you are.
Yeah, no, that's not really something I can answer. I feel like, you know, with most of the research, we just don't know that much so far about, you know, we know that nursing home residents are at high risk for all of the reasons we've discussed, but, you know, the sort of minor gradations in who's more at risk or what can be done, you know, once they're already sick, you know, I think we just don't know enough yet.
Mm-hmm. Mm-hmm. I'm just looking at, go ahead.
This is Brian. I just wanted to ask David, were there enough people in the studies you were doing with ApoE2 alleles to see if it was protective?
No. In short, no. We just didn't have the power.
I think it's definitely a lead to follow up on. It would be interesting. I think I'd love to know the basis for ApoE tracking with dementia and longevity. I don't think we really have a handle on the biology behind that at all. Yeah. Okay, so we're at the bottom of the hour, and I have a terrible fear that they're just going to shut down the technology. So I want to get a chance to thank everybody who attended this seminar. It has been recorded. It will be available on the website. I think you may be prompted to fill out a survey at some point, but that's part of modern living. Fill out a survey for anything that happens. Thank you so much to all of our 4 speakers, who I think did a terrific job. And unfortunately, with no video and no way for everyone to show their appreciation, I think you just have to accept it from me. Thank you very much.
Thank you.
Roz, I think you may have another career as a TV panel moderator if you're really good at this.
Thank you. I have a million windows open. Super duper. Thank you so much. Thank you to Trish Donato, who has done a fantastic job in setting this up. Also some heavy lifting in the back end from American Aging Association and Gerontological Society of America. If this is anything to go by, we should do more joint efforts and all come together and put aging at the forefront. It's super important in biomedical research, but also as a societal goal understand, what's going on with aging and how we can address this scientifically, whether science is popular or not in the modern era. Thank you, everybody.